Good morning. Today we're going to discuss about pharmacodynamics. In pharmacodynamics, we're going to see about the mechanism of drug action. Suppose we need to know what is meant by drug. A drug, which is a substance which act on your pathological state and also in a physiological state in the body. Yes. So, what exactly is meant by pharmacodynamic? Which means what drug does to the body and how it works. So here we're going to say about how the drug is acting on the body, where it has a specific site of action, how the effects are producing. So basically we need to know about pharmacodynamic, which is a study of the biochemical, cellular, and physiological effects of the drugs and their mechanism of action. In short, we can say it as about the drug does to the body and how do they do it. So on this thing, what are the principles? What all the drugs are exactly doing? So in per se, the drug doesn't have exactly the new effect. Okay, it always alter the pace of activity which is already existing in the body to any system or an organ or the cell. So which may called as a stimulation, which is a selective enhancement of the level of activity of a specialized cell, which is apart from the normal thing, it is always enhance the activity. Example, adrenaline, which you know, which stimulates the heart, pilocarpine stimulates the cerebral glands. The second thing we want to discuss about it is a depression. Depression, which means a selective diminution of activity of a specialized cell. It is a contrary of your stimulation. So when you have a depression, it is a selective diminution. So examples are like barbiturates, depress CNS, omeprazole, depress gastric acid secretion. Okay. So then third thing is about irritation. Irritation means it is a non-selective noxious effect and applied to the less specialized cells. Less specialized, which means it is all is called as epithelium or connective tissue. So what happens when you're exposed to certain irritants to our body? It causes an inflammation, a corrosion of the skin, which is covered by the topmost epithelial layer, which almost causes necrosis and morphological damage. Example are your ointments, whatever the analgesic ointments or povidone iodine, even though it is causing healing, it causes some erosion of the epithelial layer of the skin. Okay. The fourth principle of a drug where it is acting on is a replacement. What is a replacement means? Means we are giving something to in the normal phase. We are replacing some activity of one thing to the replace the other, which means your natural metabolites or hormones homogeneous in the deficiency state. So when you have a thyroid, for example, when you going through with the hypothyroidism, so we have to give the thyroid hormones as a L-thyroxine. Thyroxine hormone, we are giving a replacement therapy, which is all called as a replacement therapy. Example, in Parkinsonism too, we have a dopamine depletion. So we are giving a L-dopa and providing dopamine to the brain to ameliorate the signs and symptoms existing in the Parkinsonism. These are the fourth point is a replacement. Next thing is about cytotoxic action. Where you require cytotoxic action is you go through with all this is chemotherapeutic drugs. What are those chemotherapeutic drugs? Which means it goes and act on the foreign body cells and also, if possible, on your host cells. The selective cytotoxic action on the invading parasites or the cancer cell without significantly affecting the host cell is utilized for infections and neoplasms. So when you go through some fever, it may be caused through a virus or through a bacteria. So according to the diagnosis, the fever due to the bacteria or the virus, we have to choose a drug. If it's a bacterial, you have to start with the antibacterial drugs. It goes and kills the bacteria and also causes some effects on the host cells. How you choose a drug is the drug which causes a minimal effect on the host cells. And also like cyclophosphamide, it acting as an anti-cancer drugs. Since we are giving this drug to a cancer condition, it is supposed to act only on the cancer. Not. It also acts on your host cell. That is why a lot of adverse effects are existing with the chemotherapeutic drugs. So these are the five principles which are existing with your drug actions. It's a stimulation, depression, replacement, cytotoxic effect, and the topical effect. Okay. Apart from these principles of a drug, we have to move on the mechanism of drug action. Why? We need to know about a mechanism means then only you can know how where you have the drug has the effect. For the base of the mechanism, you should know about the pathology of your disease. Then only your drug peculiarly will go and act on the pathology and cause the effect. Okay. So when they have a mechanism, there is a wide loss of mechanism are there. It's through the physical action, it's through the chemical action by long ways. So when you have a simple mechanism, when you have a simple physical and chemical property of a drug, it's a handful of drug. Rest all are going with a very high mechanism, and you can see. So first one is the physical action. Physical action in the sense by the example is osmosis. Osmosis means absorption of water. So when you concern is a mannitol, it is we classify drug under the diuretics. So it is used in the cerebral edema. Second thing is absorption. Absorption means the activated charcoal. When you are giving into the poisoning conditions, this charcoal will go and absorbs the toxins. So through that of toxin, the drug poisoning affects me ameliorating. The third thing is demulcent. Demulcent is cough syrups which produces a soothing effect by coating on the inflammatory mucosa, as in cases in the pharyngitis. So these all the thing is happening with the physical action. The second thing is through the chemical action, which means antacids neutralize the gastric acid peptic ulcer, and the chelating agents, which is called as dimercator or BAR in the arsenic poison. Okay, these are the properties. What all the mechanisms through the physical and chemical action? These are all the points are coming under. The next thing is apart from your physical and chemical property, the mechanism action, and also some targets. You have a target for the drug so that the drug will go on the act on and produce an effect. Okay. So what are those targets are existing in the body where the targets we are aiming the drug. So through your development of a drug, they always speculate on the targets, whether it will be useful for the specific disease or not. Okay. The what are those targets are enzymes, transmembrane ionic channels, transporters, and the receptors. As you all know, it's a lot of molecular reactions are existing in the body through the enzymes. So either we need to enhance the activity of enzyme, as we said, there is no specific peculiar new action of the drug. It always increase or decrease the pace of the physiological activity of a body. So here enzyme, either you will enhance the activity of enzyme or it will be inhibiting the activity of the enzyme. Here, most of the drugs what we are existing here is mostly we do inhibit the enzymatic activity. So we will see how the enzyme acts and how the reaction is happening and examples around the enzymes we will be seeing. And second about the ionic channel. What are the ionic channel do you know? What are those ions? As you know, is the anion and cations. Okay. So sodium, potassium, calcium, magnesium, a lot more number of ions are existing. So these ions are passes through the cell through some channel. So sodium channel, for example, if you take, there is a sodium, it has to pass it through the channel across the cell membrane. Why? We need to maintain the homeostasis of inside and outside the cell. So this membrane, this ionic channels, if you block or if you enhance, some physiological activity will get altered, which may be beneficial for the some diseases. That is how the ionic channel means some ions will be passing through the channel. We are modulating its effectivity according to the treatment of a particular disease. The third thing will be the transporter. You had seen in the previous classes like transporters, how you transport the drug across the cell membrane. So various kinds of transporters are there. So we are focusing on the specific kinds of transporter. Okay. Fourth thing will be your receptor. So receptor, when you go, there is a vast number of receptors are there, how they are acting and what are the types of the receptor and what is necessary for what we will be seeing on the forthcoming slides. Okay. So next will be your enzyme. So enzymes are important target of drug action. I said the most important target it's concerned is enzymes. So drugs can either increase or decrease the rate of the enzymatic mediated reaction. So here increase means which will be called as an enzyme stimulation. So stimulation means stimulation of an enzyme increases its affinity for the substrate. So one thing is which is relevant to some natural metabolites only, which is called as a pyridoxine, act as a cofactor and increases the decarboxylase activity. So enzyme stimulation, it's only required for the natural metabolites only, which is a pyridoxine. Second thing, some enzymes are stimulated through the receptor or second messengers. So we see these kind of enzymes in the body and a lot of receptors also we see all the second messengers are acting. An example for this is adrenaline stimulates the hepatic glycogen phosphorylase through beta receptor. So adrenaline is a drug which stimulate HGP, hepatic glycogen phosphorylase, through beta receptor. So you have to stimulate an enzyme through a receptor to get your effect. It is called as the enzyme stimulation. So what happens with the enzyme stimulation means it goes and increases its affinity for the substrate. So rate constant, which is tagged as a Km, it is tagged as rate constant, and Vmax, which is the maximum velocity of the response. Okay. So in this kind of enzyme stimulation, we can see the rate constant of the reaction is lower. Okay. Then if you see enzyme induction. Enzyme induction means synthesis of more enzyme protein, apparently increasing the enzymatic activity. It straight away goes and increase no. In enzyme induction, when you compare with the stimulation and induction, stimulation means increase the activity of the enzyme. Here only it synthesizes more enzyme protein. Through that way, it increases the enzymatic activity. It acts so indirectly for the enzymatic activity. So that is why your Km does not change. Okay. Here the does not change. In stimulation, the Km is lower. However, Km is the concentration of the drug which is required is lower to produce a maximum response, by half maximal response. Okay. When you see a graph which shows the normal enzymatic activity and how the enzyme stimulation, induction, and inhibition with this Km and the Vmax we will see. Here, the effect of enzyme inhibition, induction, stimulation with the enzyme reactions. Okay, so we have to plot in such a way of x-axis and y-axis. X-axis goes with the substrate concentration. Y-axis goes with the drug velocity. Okay, maximum velocity of the reaction. So here, when you see the blue dot, which shows as the normal enzymatic activity, the normal enzymatic activity means you can see the purple one, which has a Km here, and the Vmax and the half Vmax. Okay, this is your normal enzymatic activity. When you see with the enzyme stimulation, where you feel always the increased enzymatic activity, which your Km is lower from the normal and Vmax is more than the normal. Okay. In enzyme induction, we see always increase in the receptor protein synthesis. Through that way, it increases enzymatic activity. It produces the Vmax will be the same as half Vmax is same as with the stimulation, and Km will be unaltered, which is same as the normal Km. In stimulation, the lowered Km as compared to the normal enzymatic activity. These are all with the positive response, like normal and in stimulation and induction. We will see about inhibition. Okay. As you know about inhibition means you go and inhibit the enzymatic activity. Okay. We have different types of enzyme inhibition, which is a competitive inhibition and non-competitive inhibition. So broadly, you have to know about a competitive and non-competitive. In competitive inhibition, you have a two type. One is the equilibrium type and non-equilibrium type. So what the competitive inhibition means? You have a substrate or a drug which acts on and goes, which is structurally similar with the normal substrate for its catalytic site of enzyme. Okay. The product is not formed or non-functional product is formed. So that is the competitive inhibition. Here, what happens with Km and Vmax is increase the Km and Vmaxes remain unchanged. The example for equilibrium type of competitive inhibition is allopurinol inhibit xanthine oxidase. Okay, you go here in a equilibrium type, increase the Km and Vmax is unchanged. Here, see in the red color one is a competitive inhibition. Km is increased than the normal enzymatic activity, and Vmax is as same, unchanged. Okay. Then we'll move on non-equilibrium type of competitive inhibition. So here, drug reacts with the same catalytic site, the substrate or the drug, whatever it is, it goes and acts on the same catalytic site of the enzyme, either from strong covalent bonds or high affinity with the enzyme. So normal substrate will not displace or inhibit. So here what happens, Km is as like seen here is Km is increased and Vmax is reduced. Okay. Here when you're concerned with the competitive inhibition, you always fixed on Km is increased and Vmax is unchanged. Okay. Then we move on to the second broad category of enzyme inhibition will be the non-competitive inhibition. In this, the inhibitor reacts with the adjacent site. When you see in the competitive inhibition, it goes and acts on the same catalytic site of the enzyme. Here, it acts on the adjacent site and not with the catalytic site. That is a wide difference which is existing between the competitive and non-competitive. It makes what it happens when it goes and combines with the non-catalytic site of the enzyme, it makes the enzyme to lose its efficacy or lose its catalytic property. Then we'll see what happens with the Km and the Vmax. The Km, which is unchanged because there is no activity with the enzyme, so it is mainly unchanged and Vmax is reduced. This is what happening with the non-competitive inhibitions. What is the example existing for the non-competitive is acetazolamide. Acetazolamide inhibit carbonic anhydrase, which is for the non-competitive inhibitor. Okay. So you clear with this four type of enzymatic activity, which is a stimulation, induction, inhibition concept. You have a competitive and non-competitive type of inhibition. So when you are seeing about the enzymatic activity, you always have to sum up with this four and this four with the Km activity and Vmax is very important. To summarize everything of enzymatic activity, when you see here stimulation, we have a Vmax is high than a normal, Km is lower, induction same as a stimulation, and Km is unchanged, which is same as the normal enzymatic activity. Competitive inhibition, equilibrium type shows unchanged Vmax and increase in the Km. Competitive inhibition of non-equilibrium type, which shows a reduced Vmax and increased Km. Non-competitive inhibition is reduced Vmax and unchanged Km. So these are the enzymatic activity. Then what is the second most target of the drug will be ionic channels. We'll see what happens with the ionic channels enzyme drug activity. So there are a lot of like transmembrane ion channels are present, which are like selective to particular ions and participate in the transmembrane signaling and regulate the intracellular ionic composition. As you know, in the normal cell, which concentration is more inside the cell and outside the cell. We have more sodium in outside the cell and inside the cell will have a more potassium. The first thing is a ligand-gated ion channel. What is that ligand-gated ion channel? Example for that is nicotinic receptor. And second type is G protein regulated channels, which are cardiac beta adrenergic receptor activated calcium channel. Third thing is voltage operated ion channel. Example, local anesthetics, which are stress voltage sensitive sodium channels. Then stress sensitive channels. Okay, these are the examples of different types of ion channels, which are ligand gated, G protein regulator, voltage operated, and stress sensitive channels. Okay. The third thing which about transporters. As you know, what are the different type of methods of drug get transport across the cell membrane. Yes, it is a passive diffusion or a specialized modes of transport. Under specialized mode, it is a carrier mediated among that active transport and facilitated diffusion. So what happens with the passive diffusion? We don't need any energy the drug to get passed through the membrane, which are lipid soluble drugs. So it get diffuses or passes the lipoidal cell membrane easily. When some drugs are having a higher problem in the diffusion, or it's going to weight more, or it's like more lipophilic or not less lipophilic, those need require some mediator to pass across the cell membrane, which is a receptor or whatever it is. So these all the transports will be focused on and will be acting in the drug. Also inhibit ongoing physiological transport of the metabolite or ion. Example, hydrochlorothiazide, which inhibits sodium chloride symporter in the early distal tubule. Okay, these are the brains of the transporters. Then fourth thing, when you look after for the targets, you see about the receptors. First, you need to know what is receptor. Macromolecule are a binding site located on the surface or inside the effector cell that serves to recognize the cell signal molecule or the drug and initiate the response to it. But itself has no other function. The receptor per se doesn't have any function, but it always serves to recognize the signal and initiate the response. Okay. So many drug receptors or proteins normally serve as the receptors for endogenous regulatory ligands or the physiological receptors. Okay. Ligand means what? Any molecule which attaches selectively to the particular receptor or sites indicates affinity or ability to bind without regard to the functional change. Okay. So what is meant by affinity? Whenever you read about the receptor, you have to make it of two factors. One is the affinity, another one is the intrinsic activity. So here in the enzyme, what you need to know the two factors is Km and the Vmax. Okay. Here, receptor affinity is confined ability to bind with the receptor is the affinity. You just have to go and bind with the receptor. So affinity of a drug for a receptor determines its concentration around the receptor required to form a certain number of drug receptor dissociations. Okay. So whenever you say the drug which has a higher affinity for the receptor, which means the same number of drug receptor dissociations will be formed even at lower concentration of the drug as compared with the another drug which has a lower affinity. Okay. So this is what it means a drug has a higher affinity. When you have an intrinsic activity means the drug which has to go and make a functional change in the receptor. So affinity means just go and bind with the receptor. Intrinsic activity after binding, you have to produce a conformational or functional change in the receptor. You have a wide range of action, which is 0 to 1. 0 is nil, 1 is a maximum. Okay. Agonist they have. So we have a different kind of activity of the receptor, which is the agonist, which is the antagonist. In under agonist, you can say is a partial agonist, inverse agonist, antagonist. So a lot other types we will be looking after with the receptor as like their the stimulation injection in the enzyme. Okay. What is meant by agonist? They have both affinity and maximum intrinsic activity. Okay. Example, adrenaline. And competitive antagonist. They have affinity but no intrinsic activity. They just go and bind. There is no functional change in the receptor and no such effect being formed as like your agonist. It is called as antagonist. Okay. Example is propranolol. What are the terms we see with the drug receptor interactions? As I say, agonist, which activate a receptor, produce the effect similar to your physiological signal module. They have both affinity and maximal intrinsic activity. Example is adrenaline, alpha and beta receptor agonist, And here you have two types of agonist here even, which is the primary agonist and allosteric agonist. What is a primary agonist means if the drug bind to the same recognization site as endogenous agonist. Okay, that is the primary agonist. Allosteric is bind to the different region on the receptor. That is allosteric agonist. Okay. Second thing is inverse agonist, an agent which activates the receptor to produce an effect in the opposite direction of the agonist, which is called as inverse. It is completely opposite direction. Have affinity but intrinsic activity with the minus n, which ranges from 0 to -1. Example is called as benzodiazepines. Okay. And antagonist. Antagonist means prevent the action of agonist is called as antagonist on a receptor or subsequent response. Here when you see with the affinity and intrinsic activity, affinity is present, no intrinsic activity, no more response will be produced. Example, you can say it is a propranolol. Okay. So antagonism is generally result from competition with the agonist. Okay. A synaptic interaction for the same site or overlapping site on the receptor, or allosteric interaction, other sites on the receptor. Okay, you have where the antagonistic drug or substance which goes and act. So accordingly, they made it as a synaptic interaction or allosteric antagonism. The another type you have is the partial agonist. Partial means what? Half. So, an agent which activate a receptor to produce the submaximal effect, but antagonizes the action of a full agonist. So, it produces only the submaximal effect. Okay. So, what happens here with the affinity and intrinsic activity is have an affinity and submaximal intrinsic activity. Example, dichloroisoproterenol. On beta-adrenergic receptor interaction, it produces a partial agonist. Okay, you clear with the types which are agonist, whatever the drug receptor interactions or relations, what you are getting us. One is agonist, as same like a physiological effect. In that primary and active we had seen, and then inverse agonist, which is opposite to the your normal, and partial agonist and submaximal effect. Then is the antagonist. Antagonist is causes no response or no effect will be found. And we had seen the comparison with the affinity and intrinsic activity. So to sum up all the effects of this receptor is here. The agonist will have affinity and intrinsic activity is maximum. Okay. Inverse agonist which has affinity, which ranges of intrinsic activity is 0 to -1. Partial agonist which have a submaximal intrinsic activity, you have affinity. Antagonist which have affinity and intrinsic activity will be 0. So among all receptor interaction, drug receptor interaction, whatever it is, there is having affinity. Only variation is existing with the intrinsic activity of a drug. Okay. So you have a lot other types. You have, so to sum up all, what are the molecular targets of a drug action is? What are those? One will be your enzymes. Under enzyme, we had seen thought about enzyme stimulation, enzyme induction, and enzyme inhibition. Under enzyme inhibition, we had seen a competitive inhibition and non-competitive inhibition. Okay. And then we had seen about a transmembrane ionic channels. We had seen a lot number of say ligand gated or ion channel, voltage gated like that. Okay. In third will be, what is that? Will be your transporters. How the drugs transport different modes, and accordingly we are acting on those transporters. And fourth will be the receptor. Under receptor, we can see about agonist, inverse agonist, partial agonist, and antagonist. So these all very basic of your mechanism of drug action, which is a primary part, which is very important when you know about the pharmacodynamic mechanism of drug action. Okay. The second thing you have to know under receptor, when you go, a lot other stuffs are there and modules are there, like two-stage receptor models and various types of receptor there, in and out. We have to know about it. Okay. So when the receptor has concern, you have a two-stage receptor model and various types of receptors. That is a very large class. I will be explaining in a minute. Thank you so much.